Okay. So as the name suggests, each place cell is spatially tuned at a specific location. So, so when the mice were running on the linear trajectory, this place cells would fire in a sequence, and we could actually decode the mice location from the place cells based on their activities because they were spatially tuned. And if you stack the place cells firing rate vector by their peak firing location, you can see that they form a nice sequence from the beginning of the spatial navigation to the end. So in the past decades of research, scientists have attempted to study the replay, pre-play during the awake and sleep states of mice, and also manipulated the place cell sequences with different tools, electrical simulation or optogenetics, that further convinces the importance of place cells as the neuronal foundations of spatial cognition and memory. So also in the recent 10 to 15 years, scientists identified time cells that fire also in the sequence during a kind of temporal navigation, which means that, for example, here in this task where Dr. Eichenbaum calls such neuronal sequences as time cells. So in this task, the mice held the memory of an object for 10 seconds in this closed chamber. And they observed a sequential activities of neurons firing from the beginning to the end of the 10 seconds maintenance phase. And such kind of cells were found in 4 mice that were involved in the study. So just like how place cells fire in sequence during spatial navigation, time cells here fire across the duration during memory maintenance. And such content-free sequence of time cells were activated irrespective of the preceding object Q. And in the recent years, Dr. Taxidis and his colleagues identified another type of sequential time cells during the memory maintenance phase. They named this order-specific sequence of time cells. For example, here, if we stack the firing rate vectors of these order A time cells, they were only presented after the offset of this order A during the maintenance of this order A memory, but they did not form a sequence after the presentation of order B during order B memory maintenance. And this suggested a new mechanism of memory retention during a working memory task. So so we know that place cells show how the brain can represent egocentric active movement during spatial navigation, and time cells, on the other hand, show how the brain can actually represent a continuous dimension during the absence of external stimulus with or without structure. So the question here is that can we actually identify time cells in the human brains during a similar working memory task during working memory maintenance? And also, if they exist, are they associated with memory performance or the memory content? So to study those questions, we leveraged a great data set shared by Dr. Rudihauser. In this data set, they recruited 21 epilepsy patients with depth electrodes implanted in the head, mainly for epileptic seizure monitoring. And during that at the time, they also got a chance to participate in this Sternberg working memory task study. So in this trial, in each trial of the task, these patients memorized a series of images, 1 to 3 images, and held that in the memory for 2.5 to 2.8 seconds. And they later got probed by a newly pop-up image that either going to be one of the images in the encoding series or not. They need to press the button to make a judgment, which would reflect memory performance. And during that task, intracranial electrodes would pick up single neuron activities from medial frontal cortex regions, including the presupplementary motor area, ventral medial prefrontal cortex, and dorsal anterior cingulate cortex, and also the medial temporal lobe region, including hippocampus and amygdala. So by applying statistical algorithm on the 902 neuronal activities, we successfully identified 157 time cells all over the brain, all over the regions that were recorded. So here are some example time cells from hippocampus and also anterior cingulate cortex. So the light blue window here highlights their time field, the time window where they were temporally tuned at. And surprisingly, the largest proportion of prefrontal motor area neurons were identified as time cells, followed by hippocampus and also anterior cingulate cortex. And if you stack the neuronal firing rates of these time cells during the during the maintenance phase of the study, you will see that they fire in a sequence, just like how the place cells fire in sequence from the beginning to the end of the spatial navigations. And if we filter out the odd trials, sorting the neurons by the peak firing rate window, time window, and then apply the same sorting to the even trials heat map, you will see that the sequence still preserves. Such kind of cross-validation shows that such sequence of time cells firing are reproducible across trials. To further understand the population activities of these time cells, we trained Bayesian decoders to decode temporal information, which is time, from the time cells population, as well as the non-time cell population, and compare that with the shuffled baseline. And it turns out that time can be best decoded from the time cell population than in the non-time cell, just like how spatial information could be decoded from the place cells. And this pattern is also true at population level if you train individual Bayesian time decoder from neurons of individuals. And then we attempted to answer the second question, whether time cells sequences are related to memory performance. We looked into some single units, and we found that most of the neurons would have a better tuning at the preferred time field in correct trials when compared with the incorrect trials, the error trials, where they made a mistake in the probe question. And if you look at the population activities from the heat map, here we downsample the correct trials in each neuron to match the incorrect trial count in the same neuron. You can see that the temporal tuning is still stronger in this correct trials, downsampled correct trials heat map than in the incorrect trials heat map. So you can still vaguely see the sequence, but the temporal tuning is just not as strong, and also some firing fields are shifting. So here, these neurons were sorted based on their time fields identified from all trials. And we quantified that by calculating the z-scored firing rate within their time field and compared between correct trials and error trials. And it turns out that there was indeed a significantly reduced temporal modulation in error trials of these time cells. To further understand what properties of the population activities were led to the phenomena we just saw, we developed a quantification method called cross correlation within single trial. So for each trial, we align each time cell based on their firing rate field, and then we compute a cross correlation between each possible pairs of neurons. And then for each trial, we would have cross correlations between time cells. So we grabbed the incorrect trials, we calculated the mean cross correlations, and because of the relatively lower number of incorrect trials in the task, we did 1,000 resampling from the correct trials populations to make these distributions. And it turns out that the mean cross correlation of incorrect trials were significantly lower than the correct trials, which means that at trial level, time cells fire in a less coordinated way in the error trials and more coordinated in correct trials. And we train test a similar Bayesian temporal information decoder model, tested on correct trials and an equal number of incorrect trials. And it turns out that there's the decoding error was significantly lower in correct trials, which means that you could decode time better in the correct trials. And if you look at the time series data, the difference in the decoding error was most significant in the first seconds into the maintenance phase. And this distinction was also true if we look at per patient level Bayesian analysis. So here, the takeaway message from this slide is that we observed a more coordinated and more reproducible time cell sequences during in trials with successful memory retrieval. Okay, and lastly, we also attempted to look for time cells that resemble the ones identified by Dr. Taxidis, so-called odor specific time cells. Here we call them Q selective time cells, which means that they are temporally tuned during the maintenance phase after the encoding of a specific particular image. So we took out we studied only the load one trials with only one image presented during the encoding phase. We successfully identified around 200 such Q-selective time cells. So here, these two cells are from the medial temporal lobe. So here they were only tuned at their field after the preferred image was encoded, but not when the other images were encoded. And surprisingly, the largest proportion of dorsal anterior cingulate cortex were identified as such kind of Q-selective time cells. And so even there's a discrepancy in the proportion of Q-selective time cells among different regions. Further decoding analysis actually showed that amygdala and hippocampus have a comparable predictive power of the preceding images. So we sorted the neurons in the same way to see how they look like in different conditions. So after encoding the preferred images, these Q-selective time cells started to form a sequence from the beginning of the maintenance phase to the end. But the sequence is not there during the non-preferred trials, where the preceding images were not the ones they were interested in. The sequence is no longer there, just like how the order A sequence cells I presented reacted differently after order A and order B. And then we also trained a supported vector machine decoder to try to decode the preceding image's identity from a population level. And it turns out that we could decode the preceding image Q reliably from the temporal tuning of this Q-selective time cells, which also suggested that their Q-selectivity were reliable and reproducible across trials. And however, such Q-selective time cell sequences was degrading in higher load when we shifted to higher load conditions. So we tried to stack the neuron in the same way to see how the sequence looked like in load 2 and load 3 conditions. And you can see that no matter where the preferred image was positioned in the encoding series, the sequential activity seemed to be gone, which suggested that there might exist other neuronal mechanism for supporting such kind of more complicated memory retention. So there might exist also combination-specific time cells that had this role, but due to the limited number of trials of different combinations, we were not able to convincingly verify that sort of hypothesis. Okay, so in summary, the takeaway message here is that we identified preliminary evidence of full existence of time cells all over the human brain, from medial frontal cortex to the medial temporal lobe regions. They encoded temporal information, just like how play cells encoded spatial information during the. So here they find sequence during an absence of external stimulus, and these sequences were found to be correlated with working memory performance. And lastly, the discovery of Q-selective time cells suggested a potential new mechanism for maintaining Q information apart from the persistent activities of other cell types like concept cells, maintenance cells, identified by Dr. Rudy Hausers and his team. And lastly, I want to deliver a great thanks to my supervisors, Dr. Valianzi, Dr. Taxidis, and also our collaborator, Dr. Rudy Hausers, for his input in methodology and his team for collecting the nice data set. And also a great thanks to my lab colleagues and also Dr. Lucia Meloni, Dr. Catherine Duncan for supporting this study. Yeah, thanks. Thank you very much.